So today I'm going to be sharing some of the information that we collected when we were trying to characterize contaminants of emerging concern, or CECs, in lake sturgeon tissues, and what some of the implications of those findings are for management. So first, I will briefly cover why we wanted to focus on lake sturgeon, and then I'll kind of get into the process of our study. And I do want to hone in here, while I'm not going to get into the specifics on the sources and fate of CECs, I do want to point out here, sorry, I do not know how it's advancing. So I'm not going to get into specifics on sources and fate of CECs, but I do want to point out that the purpose of this talk is really to highlight looking at the accumulation of CECs in organism tissues and using that as an indicator of exposure, with the caveat that just because we identify something in their tissues, that doesn't mean that while we didn't identify in tissues, that they're not exposed to that as well. And then we're using this indicator of exposure to kind of potentially get some idea of negative impacts to organism health associated with CECs. So why did we focus on lake sturgeon for this study? There's currently a very limited understanding of the biological effects in organisms associated with CEC exposure, as we're discussing today. But there is some evidence that there are physiological effects to fish and other wildlife when they are exposed to CECs. And we really have no idea what this could potentially mean for imperiled or recovering species, and whether or not these species might actually be more sensitive to exposure to CECs. Lake sturgeon are actually listed as a state endangered, threatened, or species of special concern in all Great Lakes states where they occur. They are also under consideration for federal listing right now as well. Overexploitation of this species, loss of habitat, invasive species, and exposure to contaminants have led to massive declines in lake sturgeon populations. But currently there are large recovery efforts across the Great Lakes for the species. Lake sturgeon are also unique when we think about their biology. They have a rather unusual life history for a fish. They're a really long-lived species. They can live up to 150 years. Meaning that they are potentially exposed long-term to CECs. They're also a late maturing species, and this could possibly mask some effects that we might see in growth, development, or reproduction, effects that we might see earlier on in species that are not as long-lived. They're also a migratory species of fish, meaning that they're in multiple locations and different environments, and they're potentially exposed to several different CECs. They are also benthic feeders, primarily relying on invertebrates and small fish. So if we have chemicals that are concentrating in the benthos, it's possible that we're maybe looking at really high levels of exposure through feeding. There's also currently no existing literature about CEC-associated effects in long-lived, late maturing species and the potential for population-level effects. And I will highlight for this talk, when I'm talking about population-level effects, I'm talking about impacts that occur at the individual level, things such effects associated with growth, development, behavior, reproduction, as well as mortality. And while these occur at the individual level, it's possible that these can actually lead to impacts. So the objective of our work was to characterize the CECs, and we specifically focus on pharmaceuticals and personal care products and PBDEs in adult lake sturgeon tissues, looking specifically at serum and gametes and their potential effects based on available literature in order to inform natural resource managers working to protect and conserve the species. So we captured lake sturgeon in areas across the Great Lakes where they're known to spawn. This included the Detroit River, the upper and lower Niagara Rivers, and the St. Lawrence River. Sex was determined using 3 different methods, including the direct expression of gametes, the direct observation of gonads during the surgical implantation of acoustic transmitters, as well as externally through ultrasound. Blood was drawn from the caudal vasculature and serum was extracted. Gametes, which included eggs and milk, were collected at the St. Lawrence River location only. Also need to note here that for our gametes and PBDE concentrations, only milk samples were examined for PBDE concentrations. And serum and gamete samples were submitted to SGS for determination of PPCP and PBDE concentrations. For our statistical analysis, we only focus on contaminants that were detected in at least 25% of our samples from at least one site. We first looked at the maximum concentrations of our chemicals. And then for the chemicals that occurred across all sites, we compared concentrations using a Kruskal-Wallis test, followed by a Dunn's test for pairwise comparisons. We also used a non-parametric multivariate approach to analyze this data set, and we had a few different purposes in doing this. So first off, we wanted to visually compare the differences in CEC concentrations and signatures across sites. And we did this by ordinating the data with non-metric multidimensional scaling, or NMDS. We also want to see if there were significant differences in those chemical signatures across sites. And we did this using permutational multivariate analysis of variance, or PERMANOVA, which is essentially just a MANOVA for non-normal data. And then to ensure that the differences that we were seeing across sites were actually true differences and not due to the dispersion of the data, we followed our PERMANOVA with a multivariate analog of Levine's test for homogeneity. Jumping into the results, we identified 44 different PPCPs, including antidepressants, antibiotics, and other pharmaceuticals across all sites in serum and our gamete samples. We then detected 22 different PPCPs and at least 25% of our serum samples from at least one of the sites. Benztropine, DEET, hydrocortisone, amitriptyline were detected in at least 25% of samples at each site. Then sertraline, DEET, and 10-hydroxyamitriptyline were detected in at least 25% of all gamete samples from the St. Lawrence River. We had 8 PPCPs detected in milk and 14 different PPCPs detected in our egg samples. Our PPC concentrations ranged on the low end from 0.002 to 130 parts per billion, with hydrocortisone having the highest concentrations in the upper Niagara site. Our PPCP concentrations in gametes were higher compared to our serum concentrations, ranging from 0.005 to 190 parts per billion, with diphenhydramine having the highest concentrations. The highest concentrations of benztropine, DEET, and amitriptyline were detected in all tissues from the St. Lawrence River, and these also had the highest concentrations of the antidepressant sertraline. We also noticed significant differences in our DEET concentrations in the Detroit and the St. Lawrence River. And antibiotics were more frequently detected in serum from surgeon collected from the Detroit River. And these antibiotic concentrations were also generally higher in the Detroit River as well. Then looking at our NMDS plot, we see that our samples that were collected in the Detroit River and the St. Lawrence River are clustering together. These relationships and these clusterings are primarily driven by benztropine. DEET, benztropine, I believe, is a drug used for Parkinson's disease. DEET is, of course, an insecticide used constantly in insect repellent. And then several of these chemicals over here are actually antibiotics. Then we saw a grouping of our upper and lower Niagara samples, serum samples, with these relationships primarily be driven by the antibiotic flumekine, as well as hydrocortisone. And moving on to our polybromine diphenyl ether, PBDE concentrations. Here we have plotted the total PBDE concentrations across all of our sites. Our total PBDE concentrations in serum and gametes ranged from 0.184 to 12.7 ppb and 0.08 to 0.44 ppb, again with our higher concentrations, in this case being in our serum samples. 26 PBDEs were found in serum in at least 25% of the samples at all 4 sites, and 20 PBDs were found in every serum sample at every site. 24 PBDEs were detected in 25% of our milk samples, and 14 of those were detected in all milt samples. PBDE 47 had the highest mean and median concentration in serum at each site, as well as our milt samples from the St. Lawrence River. And then when we ran an NMDS on this, there was no obvious clustering of serum and no significant difference in site or sex. So some of the implications and of our findings, other research has actually identified antidepressants, specifically sertraline, and antibiotics in the tissues of various fish species collected specifically from the Niagara River. And this might actually mean that we want to further explore possible effects related to sertraline. There is already some research on that, but maybe looking more at environmental concentrations. We know that antidepressants do have effects on fish. We know that from existing literature. We have seen evidence of predator avoidance as well as changes in reproductive behaviors for fish. Altered development, growth, and behavior in early life stages has also been observed. And what this might mean for sturgeon is it could potentially lead to vulnerability to predation or collisions with boaters, affect their ability to adequately feed, or even maybe alter their opportunities for successful reproduction. Available studies also suggest that antibiotics can influence growth, development, and reproduction and behavior in fish. This study identified derivatives of tetracycline antibiotics in lake sturgeon tissues, but data are lacking regarding the effects of antibiotic metabolites and degradants. And I will note here that, we're not really sure if the sturgeon were exposed to the actual metabolite or the parent compound and actually, metabolized it themselves. We have no way of knowing that, but there is very little data regarding specific effects, metabolites or degradants. Some other things to think about seasonal variability. So, our data really is only reflective of what's going on at a specific point in time. It's possible that during different parts of the year, lake sturgeon may be exposed to different chemicals.
3 minutes, Amber.
Also point out that benztropine and DEET were detected in tissues at all sites, and we have very limited, if any, information about the effects. And then thinking about the distribution of PPCPs in lake sturgeon tissues, we noticed differences in the types of chemicals in the different tissues, including serum and gametes. For example, sertraline was detected in more than 85% of the gamete samples and in less than 10% of our serum samples. And 10-hydroxyamitriptyline was detected in eggs, but not in serum. Contaminants detected in gametes could be indicators of exposure and effects to developing embryo and larva. And while we did not calculate bioaccumulation factors in this study, it could help with identifying potentially the fate and effects of antidepressants and antibiotics in fish tissues and organs. Moving on to implications of our PBDE findings, we know that PBDs are still quite prevalent in the Great Lakes. This data shown here is looking at total PBD concentrations from, I believe, 2002 to 2016 from EPA surveillance program. And we see that, while concentrations do fluctuate, PBDs are still prevalent in fish tissue. We know that exposure to PBDEs could lead to reproductive, endocrine, developmental, and behavioral effects of mortality. And specifically for sturgeon, we're concerned about the disruption of thyroid hormone and the effects that could have on lake sturgeon reproduction. And my colleague, Steph Hummel, will get into that. So what are some of the management implications? Areas with known sources of PPCPs and PBDEs, for example, near wastewater treatment plants, are maybe not ideal for stream-side rearing facilities, spawning reefs, or reintroduction programs. And if biological effects are observed in lake sturgeon or there is limited success with recovery efforts, managers might want to consider the potential. And there are lots of people to thank that made this work possible. And with that, I will take questions if there is time.
Great, thank you, Amber. I think we have time for one question.
Were there any hospitals or residential medical facilities within the drainage area of your site, or are these concentrations common in the literature?
We sampled across several different urban areas, and I guarantee there are probably some inputs from those various sources in these areas just based on where we're sampling. That would definitely be I think, interesting to further explore though.